Hi everyone. In this video, we're going to be talking about the three distinct processes that allow for urine formation. The first one is filtration. Filtration by definition is hydrostatic pressure forcing water across a filtration membrane. And in our case, we're going to be talking about the renal corpuscle. So that would be the glomerulus and Bowman's capsule. So first, let's talk about the different things that we would find in the blood and in the blood capillaries. So we know that we have about 55% plasma and about 45% formed elements. So things that we're going to find include water, small solutes such as ions, sodium chloride, calcium, things like that, glucose, amino acids, certainly other things like hormones, and then some larger structures like proteins. So all kinds of proteins, albumin, transport proteins, immunoglobulins, things like that. And then 45% is formed elements, of which 99.9% is red blood cells. So we also are going to find big cells like red blood cells, for example. Now, also, because we're talking about the kidneys, we need to address the different waste products that are going to be passing through the blood vessels. So the three waste products that we're interested in are urea, which is often from breakdown of amino acids, uric acid, which could be from breakdown and recycling of RNA, and creatinine, which can be from breakdown of creatine phosphate, which is basically an energy storage sort of capacity in the in skeletal muscle. So as creatine phosphate breaks down, it makes creatinine, making our third waste product. So let's just consider the glomerulus because the difference between most of the other capillaries that we discuss and the glomerulus is that this is a fenestrated capillary, meaning that there are large holes in the capillary. Now, if we think about the job of the kidneys, what we want to be able to do is extract all of those waste products from the blood. We don't want them in the blood. We want them to be eliminated. Now, if we wanted to move them across the membrane, taking into consideration our other membrane transport options, it really is just not, it's not doable and it's not efficient to do it any other way. So, for example, the waste products are too big to squeeze in between cells. The waste products are too big to use channel-mediated diffusion or carrier-mediated diffusion or transport. And we don't want to expend any energy because it would be unbelievably energy costly. So that sort of takes any type of active transport, which includes primary active transport, which is probably too big for anyway, and exocytosis off the table. So we need to find a way to remove the waste products efficiently. And so basically, if we make the pores in the glomerulus big enough for the waste products to fit through, then the waste products, as they pass through the glomerulus, will easily just move through those pores and then sort of fall into Bowman's capsule. So the thing that makes it slightly complicated is that if we think about the waste products as being kind of big, but in relation to the other things, bigger than the ions and small solutes in water, but smaller than the proteins and the red blood cells, we have to make the pores just big enough for the waste products, which means that anything smaller is also going to move. So the stuff that moves from the glomerulus into Bowman's capsule to make filtrate is going to be the water, the small solutes, and waste products. And then the stuff that doesn't move is going to be things like red blood cells and proteins because the holes are not big enough for them to pass through, which is important. We don't want them to be that big because then we wouldn't be able to filter. So think of this as the first sort of step in picking and choosing what's going to move. And we're going to lose some extra stuff that we don't want to lose, but we're not going to lose everything. So this process only occurs between the glomerulus and Bowman's capsule. It does not occur anywhere else in the renal tubule. And that's important because if it did, then our process just wouldn't come together appropriately. Also, because we mentioned that we don't want a process that's too energy costly, just for the sake of comparison to the other two components, filtration is entirely passive. It is only passive. It does not require any ATP. All right, now let's compare that to the second process, which is reabsorption. Reabsorption means removal of water, ions, and nutrients from the filtrate back into circulation via the peritubular capillaries. And this process can be passive or active. So here's our renal tubule, and the renal tubule on either side is lined by renal epithelial cells. And we'll talk about the specific types of cells in the different segments of the renal tubule, but for the purpose of reabsorption, we just want to sort of identify that it's a single layer of cells that that sits around the renal tubule. So these cells are going to display polarity as many epithelial cells. The side that's facing the renal tubule is the apical side, and the side that's facing the ECF is the basolateral side. So on the side of the basolateral side of the cell, we're going to have interstitial space, right, filled with interstitial fluid, and then we will have the peritubular capillary. So the peritubular capillary, think of it as there's plenty of capillary for all of the renal tubule. And even though the first drawing didn't necessarily depict this, it's in very close proximity to the renal epithelial cells. So it's not an incredible distance. So we know that as we move the waste products into the renal tubule, so into Bowman's capsule, and then it will just naturally kind of follow into the renal tubule. We have all these other things in there like water and ions and glucose and amino acids that we didn't actually want to get rid of. We want to save everything except for the waste, and not necessarily every bit of it, but most of it. Because if we were to just let everything sort of leave all at the same time, we would suffer immense dehydration in less than a day. So it would be a very, very rapid decline. So we need to save most of that water. So basically what we want to do is we want to use the other transport mechanisms that we know. So things like use of aquaporins, things like use of carrier-mediated transport and channel-mediated diffusion, and primary active transport, secondary active transport, all those things. And we're going to move the stuff that's in the renal tubule that we want to save, right? Water, ions, small solutes like glucose and amino acids, back into the renal epithelial cells. So that means that it has to cross both the apical surface and the basolateral surface. It then will move into the interstitial space, and then from the interstitial space into the peritubular capillary. So it's not enough to just get it into the renal epithelial cell, because it would just accumulate, and eventually there would be too much, and it would be either there wouldn't be a gradient, in which case anything that's passive just wouldn't function, or if it's active, it just would fill up the cell, and the cell would just not be functional. Maybe it would burst. I don't know. Something just that doesn't work. So it doesn't count if we only get it into the renal epithelial cell. We need to get it into the peritubular capillary. So the thing to keep in mind about reabsorption, especially as we contrast it with secretion, is that the direction of movement is the critical piece. From the renal tubule back into the peritubular capillary, which means that it's being saved for the body. So if it gets back into the peritubular capillary, that means that ultimately it's going to leave the kidney through the renal vein and be part of general circulation again. So this process does not happen in Bowman's capsule, but it does happen in all of the parts of the renal tubule. So the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. All right, so now we're going to compare that to secretion. So secretion is the other process that sort of is the opposite of reabsorption. So secretion refers to transport of solutes out of peritubular capillaries into the filtrate, and this also can be active or passive. So again, we can take into consideration all of our membrane transport mechanisms, and we're essentially going to draw the same picture that we just drew, but I'm going to add a few things. So in blue is Bowman's capsule, and then the renal tubule in black with the green cells. And then we have our glomerulus, efferent arteriole, and our peritubular capillary. So in this instance, let's think about maybe like a toxin that we wanted to get rid of that didn't actually filter in the glomerulus. So that for whatever reason it was able to, and to be fair, not every single droplet of water, not every single molecule of glucose falls out of those pores in the glomerulus. Some of the things will keep moving. We just lose a lot of it. So it's entirely believable that something like a toxin or some other thing that we wanted to get rid of, that we didn't get rid of enough in the glomerulus, we can still eliminate. So sort of imagine that this toxin is going to travel, through the glomerulus, through the efferent arteriole, and now it's in the peritubular capillary, and the body says, I want to get rid of this. So that the toxin is going to move from the peritubular capillary into the interstitial space, into the renal epithelial cell, and then into the renal tubule. And once it's in the renal tubule, it has the ability to be eliminated. So it's going. So the critical difference here between secretion and reabsorption is that the direction of movement in secretion is from the body. So we originally were going to save it, right? It didn't get filtered out in the glomerulus, but it was in the peritubular capillary. So in the peritubular capillary, we say that it has kind of one of two choices. It's either going to keep going, in which case it'll leave the kidney through the renal vein, or it's going to be secreted into the interstitial space, in which case it will ultimately arrive in the renal tubule, and then it will be eliminated. So if it travels all the way through the collecting duct, it will end up in the renal pelvis and then the ureters, and then it will be eliminated, and so on. So this process of secretion can occur in the proximal convoluted tubule, the distal convoluted tubule, and the collecting duct. So that is that's all for the processes that allow for urine formation. And as we move forward, we're going to be talking about each segment very specifically. So Bowman's capsule, then the proximal convoluted tubule. And we'll talk about which stuff is moving in which place and exactly what transport mechanisms are available to make that happen. And then also how things like hormones affect what we do and don't eliminate. So I hope you enjoyed it. Thanks for watching.